Hello everybody, it's Biology Sunday again. Okay, now we are on A3.2 classification and characteristic. Okay, classification is the old thing. Okay, characteristic is the new thing. Okay, so it's a change, a big change. Okay, paradigm shift from classification to characteristic. Okay, see what happened. It is AHL level, so a little bit conceptual. Okay, so get ready. So first of all, we learn about naming a living organism. Okay, we learned it before, right? Two-name system, genus and species. Okay, and in the world there is a immense diversity of species and lots of species. So we always need a classification to distinguish and group different species. And after classification is completed, and we can do a further study on them. Okay. And then, for example, we have the plant kingdom and the animal kingdom. Okay, this is traditional hierarchy of taxa. Okay, traditionally there are six rank. Okay, kingdom, phylum, class, order, family, genus, and species. And there are five kingdoms. Okay, animal, plants, fungi, protists, and monero. And we will study a little bit about the animal and the plant. You don't need to learn. This is a little bit the hierarchy solving. Okay, so you need to learn about them in detail. Just have a rough understanding is excellent. For example, the classification of the plant kingdom, there are lots of different different of phylum. Okay, there's a five. Okay, Bryophyta, Phytocinophyta, Coniferophyta, and Geospermophyta. Okay, and then they have different appearance. Okay, for the Bryophyta, it actually is most sensitive. Okay, for the Phytocinophyta, it's really fern. Okay, Coniferophyta is a little shaped pine leaves. Okay, and Angiospermophyta is a flowering plant. Okay, and then there's there's a good structure for the last three. Okay, and the first one has no vesicle tissue, no stem, or no woods or no leaves. Okay. And next one I want to as some mere understanding about classification of animal kingdom. Animal kingdom are classified into different also. Okay, Porifera, Cnidaria, Platyhemifera, Annelida, and Mollusca, Amphipoda, and Chordata. Okay, actually, lots of them are not concerned by us. Okay, but we have to learn about them a little bit, just a little bit. Okay, for example, the Porifera is a sac-like spongy body. Okay, low symmetry, low organ, very basic thing. Okay, Cnidaria, okay, has a soft body. Okay, has a stinging tentacle. Okay, radially symmetric. Okay, has mouth but no anus. Okay, Pyraminifer and smooth flat worm or a smooth unsegmented body. Okay, and the leader, okay, is a worm-like body. Okay, segmented body. And mollusca, okay, and this is an unsegmented body and it have a shell. Arthropoda is a smooth segmented body. Okay, and then it has an exoskeleton just like the insect. Okay, and the chordata is okay, it is all the vertebrae and all the invertebrate is a chordata. Okay, so we are chordata. So next one we talk about a little bit about vertebrates because we belong to it. We need to have understanding of it. Because if you want to know in junior form, okay, we learn about five vertebrates, five classes. Okay, fish, amphibians, reptiles, birds, and mammals. More familiar works, right? Daily life. Okay, and you look at the characteristic of them. Can you find the distinct feature of each of them? Yes, I list out here. For the fish, it is fin. Okay. Amphibian is a skin that respirates. For reptile, it's a dry skin with scale. And for birds, it's a feather. For mammals, for us, we have hair, fur, mammary glands to give birth to young, and then feed young with milk. It is us. Okay. And then eukaryote, okay, bigger thing. And then all organisms are classified into 8-level hierarchy of taxes. Okay, from the small to the big one. Okay. And there are new one called domain and kingdom. Okay, and all organisms are classified into three domains: the bacteria, archaea, and then eukaryote. For two organisms with the same taxes, okay, just remember branch them out. Okay, if they have the same taxa, and then they must have the same taxa above it, but they may have different taxes below it. On the contrary, with a different taxes, okay, they must have a different taxes below it, but they may have the same taxa above it. Okay, just think about the spreading out of the taxonomy, the diagram, and then you get it. In IBDP, okay, the order of taxes asked may be reversed. Be careful. You need to remember the sequence. Okay? Sometimes they are reversed. Okay, species, genus, family, order, class, phylum, kingdom, and domain, from small to large. So be careful. And there's a problem of traditional hierarchy of the taxes. This is a paradigm shift for you. Before we just look at the appearance, okay, outside, okay, of species of organism to separate them, okay. But actually nowadays we think that the evolution is more important, okay. Divergence from evolution is very more important to sort out the relationship between the organisms. So now we use evolution as a basis. And how can we find out the evolution? We'll talk about it later. Okay, so this kind of classification by evolution, we call it a Cladistics. Okay, and the species will be joined with the same characteristic. We joined it with class. Okay, so it is a big change from Cladistics to the taxes. Okay, and the concept is by this evolutionary relationship. Okay, we are talking about do they evolve from a common ancestor? If they evolve from a common ancestor very closely, then they are closely related. And if they have they don't have a common ancestor, then they are very divergent. They are not very related. This is the concept of the evolution from a common ancestor, close or far away. So this kind of a natural, we call it natural classification. Okay. This is some advantages. Okay, so disadvantage is identify a species by natural classification. We identify the evolutionary path. Okay, and then we close evolutionary relationship and a common interest that are related to this natural classification. So after we do this classification, and then we know the evolutionary relationship between them, and this kind of natural classification is a cross-cultural internationally accepted language. Okay, all the world use the same classification according to this evolutionary relationship. So we have a common language. Class. Okay, we talk about class. So class are groups of organisms. Okay, just like species. Now we call it class. Okay, as organisms with common ancestor and shared characteristics. Both things. Okay. And then we need to find out some evidence, okay, why we say that they come from the same class, why they have the same common interest, shared characteristics come from I'm not sure about evidence. We talk about the evidence later. Evidence, yeah. For example, having a homologous structures, similar structures, okay, and the base sequence of a genes are similar, and all the protein produced by them, that the amino acid sequence are similar. Both are evidence. Okay. For example, when we do all this investigation after getting the evidence, we can draw the cladogram. Cladogram shows they starting from this common ancestor at the bottom. But this common ancestor is always doesn't know who are they, so they are not named. Okay, named. And this is empty. If this is if we know who they are, then we'll write the name there. Okay, so these two common ancestors are too far away. We don't know who are they, so it is empty. When they spread out into different clades, okay, if they are close, they are divergent from the common ancestor. For example, A and B are divergent from the common ancestor X, so A and B for a clade. But C and D are divergent from different ancestor, right? So they are not doesn't form a clade. And sometimes if in the cladogram there is this one MY, a million years ago, there's a time scale. Okay, it means that the short and the long distance reflects the time between divergence. Okay, the short time is long time, but sometimes they don't have this time scale. Okay? If there's no time scale here, it means that this length has no meaning. Okay, it can have two kinds of drawing. Okay. And then we can talk about it later again, in details, okay. And then yeah. Okay, two formats, okay, left and right, okay. And then all organisms in the same class are most closely related to each other. A, B are closely related to each other. C, D, no. And E, F are closely related to each other from the same divergent point, okay. And then mutation rate. Mutation rate is evidence, okay, because we assume that the mutation rates of DNA and the protein's amino acid sequence is very constant. We assume it is constant, just like a molecular clock, okay. Maybe 10 years one or 5 years once mutation, okay, for different species, okay. So by looking at that they have a different mutation, the number of mutation, we know that they are divergent at different times. Just basically it's a more mutation, more number of mutation, they have been divergent from a far away common ancestor. But actually, it's an estimation. Mutation rate depends on another thing, right? The length of the generation, the size of population, the intensity of a selective pressure, and other factors, right? So this assumption of a constant mutation rate is a little bit not accurate. So although I write it here, DNA mutation is found to happen at a relatively constant rate, but actually it's not. Okay, I'm sorry. Okay, so when we find out different organism, we can compare the amino acid base sequence. Okay, the differences. Okay, the more the differences, the longer for the divergence. Okay, but first of all, look at them. They may come from difference. Okay, so this annelida and then the mollusca are completely different. So C, A, and C can be eliminated. We don't need to care about C. Okay, completely different thing. Okay, so we talk about A, B, D, and E. Okay, and you compare them. Obviously, A and E are the smallest number of amino acid difference. Right? Smallest number of mutation. Of course, they are closest together, and the farthest one is A and C. So when you draw the graph, A and E should be close, and then A, B, and then A and C. Okay, and then the D is even far away. And we have the other basis for constructed cladogram. So for example, the mutation rate. Okay, for the mutation rate, we can look at the DNA or the RNA. Okay, the base sequence of the DNA or RNA. But this one you have to extract the DNA and RNA from the organism. Okay, then you have an easier way. You can look at the amino acid of the protein produced by them. Easier, they produce the protein when you just get the protein and investigate the protein. But the problem of this easier way has a problem because different triplet of bases can produce the same amino acid. So the same amino acid sequence doesn't mean that the bases are actually really, the DNA are really have the same sequence, right? So it is an easier way, but not accurate. The DNA is the accurate way now. And of course now then getting the genome is easier, right? Cheaper. So we rely on this thing nowadays, okay? And then by this, it is we call it a molecular evidence. Molecular evidence is the base sequence of the DNA and RNA similarity, and then the protein, the amino acid sequence of the proteins. Okay, we call it molecular evidence. So not the outside, not the appearance. This is the molecular way. Maybe they have different appearance, or maybe they have similar appearance, but in the molecular level, okay, DNA, proteins, maybe they have lots of mutation, lots of differences. Okay. So it's sequence of divergence. When you write this thing, it's in the cladogram. Remember, it is by the molecular evidence, not by the outlooks, okay, not by the appearance, okay. So X, Y, Z to Z, C and D are closest, okay. Z is the last divergence. X is the first one, okay. But how do you know X, Y, Z? Okay, we have four things A, B, C, D. So it is a guessing game, okay. You are just like a detective. For example, we got PQRS, okay. We have four organisms. We have the sequence, okay, gene sequence. Okay, here, okay, we look at here. First of all, you recognize something, they are identical. Okay, T, C, G are identical for three organisms. Okay, only the P has GGT. Okay, so we think that P is the first mutation. Mutation X, P is split out. Okay, and then look at the other three. Okay, and then the QRS, you look at something common just to have it, and the third one that's CCA. CCG. So Q is the second mutation's result. Okay, and look at the last one. I make it wrong. Okay, so whether C and G are different. Okay, but which one is a mutation? Look at before. Okay, GGGC. Okay, so C should be a mutation. Okay, I'm sorry, I should shade this one like that. S will split out. Okay, and the R is already so mutation X, Y, Z. X is the first, and the Y is the second, and then Z will be the last one. Okay, so when we join, how can we join? We join by different node, branch, and whatever. So we go, and then we find it out. This is a guessing game, right? We have a theory, right? It's just different criteria for judging it. Okay, maybe the real world is not so clear cut. Okay, so maybe by the same methodology, maybe we come up with different cladograms, right? Yeah, with different focus, okay, on the DNA sequence or on the amino acid sequence, okay, different parts, right? So maybe we come up with different cladograms. So this is a parsimonial analysis, is the comparison of different cladograms, okay, to see which one is the most probable one. The concept is they require the smallest number of sequence changes the best, right? This is the trick, okay? This is the criterion, okay? So just remember, parsimonial analysis is the comparison of different possible cladograms to find out the most probable one, okay? And then when we draw the cladogram, we have different technical terms. Okay? We call it root, loop, and branch. The root is the first divergent point, okay? The root, okay? And the loop is a divergent point, okay? Any branching out point, we call it a loop, okay? And the branch is a path that has no diverging point, okay? And the terminal branch is the last branch that is connected to an existing organism. So every existing organism, Homo sapien or whatever, okay, is connected by a terminal branches, okay? So it is just a structure, okay? How to name them: root, loop, and branch. You need to remember this technical there, okay? And there's an evaluation and classification by Cladistics, okay? So we classify because we rely on evidence, right? When the theory evolve and we have more fossil coming out, new knowledge on the common adjuster, evolution path or evolution relationship, okay, when this thing change, okay, we have more evidence, so the cladogram can change, right? Yes, can change. So it is more dynamic nowadays because we are not looking at something that is outlooking is about inner, okay? So about the DNA, about the base sequence, about the genotype, okay, nowadays. So it is more inner. So we have more flexibility, so I say, or more evidence might come out, okay, unexpected. And we have another example. For example, for giraffe, okay, all giraffes look the same, but actually they have come from 4 different divergent points, okay. So they have 4 separate species for the fishworm family, and then we think that they're just one, okay, but actually there are 6, okay, families, okay. But why we guess they are not one? Okay, this is the clue here, okay, because of the new information, maybe a new ancestor or new evolution path, or and then it will change. Okay, for example, for the fish, okay, why we think that it is not one family? Because we have one thing lack. Okay, they have a lack of a defining trait. Okay, trait. We can't. They don't have something common. Okay? They don't have a really distinct feature for this fishworm family, although they look a little bit similar. So we think that they might come from a different scale, okay? So we study the DNA of them, okay? We don't study DNA of everything, okay? We found out something that's interesting, okay? For this one, they don't have a common distinct feature, so we study it, and we found out yes, they come from 6 different families, okay? It's a breakthrough, okay? So in examination, student, you may use the common names or the scientific names to refer to an organism. So if you remember it's a common names or you remember it's an organism, technical names or scientific names, you can use them. Okay, we go back here. Okay, for the 3-domain system, in 1977, we studied the rRNA of the all organism. Okay, they are classified into 3 domains. Actually, before it's 2, prokaryote and eukaryote. You remember eukaryote and prokaryote, and then the prokaryote is further separate into bacteria and then archaea. They separate into 2. Okay, archaea is found in extreme environment, and bacteria is found in normal environment surrounding us. Okay, so this is a 3-domain system that we are using nowadays. And then 3 domains has different features, so you don't need to exactly remember them. Okay, it's hard to remember. It is a facts, okay, from there you can Google it easily. So maybe in examination they will show you this one, and then you have to discuss or distinguish between them, okay. And another technical term was the intron, was a lipid in the cell, in membrane, and then a protein associated with DNA was histone. We'll learn it very soon, okay. Are you excited? We'll learn it very soon. And thank you, okay, for this one. Is a little bit fun here. It's just a change from Texas to Cladistics. Okay, so a lot of new terms. Okay, so the old thing about Texas, the phylum of plants, okay, the phylum of animal now is a little bit fake. Okay, and other thing is quite factual about them. So to this one, I think IBDP has changed to more research type, okay, more analytical, more reading comprehension, just like, okay, we read the cladogram and you try to understand what happened. You need to describe it, okay, look at the features, okay, whether it has a time line on the right hand side or not, okay, this is quite modern, okay. So that's it, okay. If you like our sharing, remember subscribe and share it with, well, your friends, okay. See you next Sunday. Bye.